right, so good morning, everybody. So we've got the fourth unit here is the molecular epidemiology of antibiotic resistance. And I come at this a slightly askew angle because I'm by training an environmental microbiologist who has been getting increasingly involved in trying to understand the ecology of antibiotic resistance. And so as I was working to prepare this talk, I wanted to share a little bit about the antimicrobial resistance problem, and also how it's a little bit different from, more straight from simply infectious disease epidemiology. So, as a reminder of what we're looking at, antimicrobial resistance, this is pulled out of the CDC Antimicrobial Resistance Threat Report from 2019. They were estimating nearly 3 million infections per year with antimicrobial resistant bacteria, antimicrobial resistant bacteria and fungi. They like to include C. difficile as related to antibiotic resistance use, although it's a slightly different issue. And then over 35,000 deaths per year in the United States alone with antimicrobial resistant due to antimicrobial resistant infections. And that doesn't take into account the many people who have just prolonged infections, increased complications, increased healthcare costs for antimicrobial resistant infections that stop short of death. And at this point, I don't know, I think the majority of us likely have experienced at least one infection where the first round of antibiotics they tried on it has not worked. I know that, personally as a parent of young children, we've had that very regularly with ear infections. Quite a few infections these days may not respond to the first round of antibiotics tried. And, another way of looking at this problem, you may have seen this timeline before, where it's a timeline of when major antibiotics were introduced, and then how quickly the first resistant infections were identified. And as you can see, antibiotic resistance is not a new problem. From the very beginning, from the very first antibiotics we found, they sooner or later, once the use of those antibiotics becomes widespread, you start seeing resistant bacteria pop up. But in some ways, the concern is that the timelines are perhaps getting shorter, where a lot of the newest antibiotics, they'll have resistant isolates popping it up within a year or less of introduction. And the other concern is that by some measures, the number of new antibiotics that we've been bringing out has slowed down a bit. So why is that? Well, one of the problems in terms of designing new antibiotics is that in order for an antibiotic to be effective, it has to, one, target something that's really critical to cellular function so that it's hard to work around it. But at the same time, it needs to be something that is both central to the cellular function and also sufficiently different in your target organism from eukaryotes so that you're reasonably certain that your antibiotic isn't also going to kill your host cell, right? And so that's part of why fungicides in particular are even more difficult to develop new antifungals that can be used in humans than even antibiotics, because we've got so much more cellular machinery in common. And so essentially nearly all antibiotics out there target one of about 5 different cellular processes. We have antibiotics that target cell wall formation, DNA and RNA synthesis, folate synthesis or other components of sort of central metabolism, cell membrane formation, and protein synthesis in the ribosome. And that's basically the set of targets you have. On the other hand, when bacteria are trying to evade antibiotics, you have multiple mechanisms to get around any one of these antibiotics. So one of the simpler ones is to have efflux. You just keep the antibiotics from getting into the cell or pump them out faster than they can get in there and cause trouble. You might have immunity where the bacteria simply start out with, for example, a ribosome that the antibiotic can't target, or bypass where you sort of wire metabolism around whatever step the antimicrobial is targeting. You have target modification where you simply change your target enzyme enough that the antibiotic can no longer target it. Or you can produce inactivating enzymes that might break down the antibiotic before it can get in there and interfere with the cell function. And as you can see, a lot of these processes are listed in multiple resistance types, so that there's more than one way to get around any one antibiotic. And then on top of that, the vast majority of antibiotics, at least the original precursors, are based on natural products that were initially invented by bacteria and fungi as part of natural bacterial warfare. We've all heard the story of penicillin's discovery, where it's an antibiotic that's produced by fungi in order to reduce competition from bacteria. And that's part of why antibiotic resistance can move so quickly, because there is already resistance mechanisms out there. It's a matter of getting those from the bacteria that might already have developed these resistance mechanisms but aren't necessarily pathogens. Can these resistant mechanisms get into the pathogens, or do the pathogens, invent them de novo? And so in general, the other problem with antibiotic resistance is that antibiotics naturally exert very strong selective pressure for resistant bacteria. You have existing mutations in the population that might make the bacteria more or less resistant to the antibiotics. When you apply the antibiotics, you very successfully kill off all the bacteria that are not resistant, and then the resistant bacteria multiply. And then even in the absence of the antibiotics, unless there is a very strong negative pressure against the particular resistance mechanism you have, it tends to have the population tends to have a much slower reversion rate. So the resistant bacteria will not become a smaller fraction of the population unless the particular resistance mechanism has a strong negative effect on fitness in the absence of antibiotics. And unfortunately, in many cases, we have found that on typical timescales, this reversion of the population is very frequently kind of slow. And then on top of that, when you're taking systemic antibiotics to target an infection, you're not just applying this pressure to the particular pathogen you're targeting, but to all of the bacteria in the entire human body, which means that you can also be encouraging the growth of resistance in off-target populations as well as your pathogen of interest. And then we have another problem, which is that bacteria in particular are very good at exchanging genes. Genetic material moves between bacterial species with a pretty high frequency, which means that antibiotic resistance just doesn't stay in one bacterium. It can move from bacterium to bacterium. And as an example of why we need to consider horizontal gene transfer whenever we think about antibiotic resistance epidemiology, I've just went through the CDC major antibiotic resistance threats report and marked with a star here all of them where resistance is associated with mobile genetic elements. And as you can see, it's the vast majority of these antibiotic resistance threats. You have to also consider acquired resistance and horizontal gene transfer rather than simply innate spontaneous resistance. So when you're thinking about horizontal gene transfer, there's three major mechanisms by which genetic material moves between bacterial cells. You have transformation, which is one of those processes that a select few organisms, not all bacteria can do this, but the ones that do it tend to be very good at it, which is that they can bring in foreign DNA from the environment and then incorporate it into their chromosome by homologous recombination. And naturally transformable bacteria, such as Acinetobacter bailii, tend to be very good at acquiring antibiotic resistance. Another mechanism that's very common is conjugation, where this is typically associated with, for example, sex plasmids, where you have plasmids that carry with them the machinery produce a pilus that will hook onto another bacterial cell and facilitate the transfer of a plasmid from one bacterium to another. Or if this plasmid gets incorporated into the chromosome, it could also facilitate transfer of chromosomal DNA across organisms. And then we also have transduction, where if the bacteria have an active bacteriophage infection, as part of the final burst of lytic bacteriophage, they tend to produce a large number of phage, package them up quickly, explode the cell, and send them off to go attack other bacteria. And sometimes as part of that, rather than just packaging phage DNA into the genome, you might accidentally package up some chromosomal DNA. And then you get sort of pseudophages that can then transfer DNA into other bacteria. And so all of these things allow, for example, an antimicrobial resistance gene to move from one bacterial lineage to another, independently of the evolutionary history of that bacterial lineage. And then on top of that, once the genetic material is within the cell, it doesn't tend to stay put. So transposons or insertion sequences can facilitate the movement of, for example, antibiotic resistance genes between plasmids and the bacterial chromosome, between prophages in the bacterial chromosome, and then back out into new plasmids, or between two different plasmids that might co-occupy a single bacterial cell. And so all of this together means that antimicrobial resistance has a tendency to not just stay put, but can move among bacteria, which adds to the complications of the fact that whenever you impose antibiotics, you select for antimicrobial resistant bacteria across the entire host system. Now, we do have a few saving graces. Horizontal gene transfer has a few limitations. That means that it tends to be most common in more closely related organisms. And the less closely related the organisms are, the less likely a gene is going to successfully make a jump between those organisms. This can be for a few reasons, one of which is that if the gene is going to cause resistance once it's in the new host, it needs to be expressed. So that new host's cellular machinery has to be willing to produce that protein. Once it's produced, it needs to work properly in the context of the new cell. And then on top of that, bacteria, while they like to have horizontal gene transfer for the ability to acquire new capabilities, there's also some major downsides to being a little too flexible in your core genetic material. So in general, bacteria have a lot of immune systems that are established to prevent the entry of phages that can also limit acquisition of foreign genetic material that can include like CRISPR-Cas systems, restriction modification systems, and similar. And then on top of that, if your antibiotic resistance gene is on a plasmid, if the plasmid is going to stick around for any time, the replication machinery for the plasmid needs to be compatible with the host cell, which again tends to be most common among closely related organisms, although depending on the plasmid type, they might have broader or narrower host ranges. All right, and so then the last component that you need to consider when you think about the epidemiology of antibiotic resistance is that the vast majority of antibiotic resistance threats are also organisms that have substantial either environmental populations, commensal populations, or close relatives that are commensal, or can have asymptomatic colonization, which adds to the side selection where you apply an antibiotic and then that selects for resistant strains, even if they aren't necessarily the organisms that you're targeting with that particular antibiotic at the time. And again, I went through and marked here all of these antibiotic resistance threats that have either environmental populations such as Aspergillus or commensal or asymptomatic populations that are involved in the spread of these organisms. And that can increase the frequency with which they, one, encounter antimicrobial resistance, and two, encounter other organisms that might be carrying resistance genes that they could then acquire and then apply in the context of resisting treatment as an infection. And so that's given rise to the increasing interest in sort of One Health, where we need to consider when we think about slowing down the spread of antimicrobial resistance, we don't just look at hospital spread, but we also have to consider the role of spread of antibiotics in the environment and antimicrobial resistance. So we might have antibiotic usage in individuals that then select for resistance in all of the different organisms across their body. We have concerns where the resistant bacteria that are produced then enter the wastewater treatment system, where they can either be incompletely killed before release, or you might have wastewater leaks that then result in those bacteria escaping into the environment, exchanging genes with bacteria in the environment. We have agricultural use of antibiotics, which then could result in selection for antimicrobial resistant bacteria that either enter the environment and interact with human pathogens, or might be carried on food, ingested by individuals, and then enter their body, where they can either, if they are food-related infections, cause direct infections, or potentially exchange genes with pathogens already present in the individual's body. And so these are all processes that have been observed, and we're still working out how much each of these individual processes might be increasing the spread of antimicrobial resistance in general. But as I said, many of the major threats are organisms that have environmental or commensal populations that might be participating in this spread outside of a clinical context. And so part of a picture of how this can interface with horizontal gene transfer, for example, is that you might start out with an environmental bacterium carrying an antimicrobial resistance gene on their chromosome that was that was developed initially to help defeat natural competitors that produce antibiotics. But then you can get that gene onto a transposon where it can move into a plasmid that can then be transferred between, for example, an environmental bacteria or a host and a host-associated bacteria. Or maybe that bacterium is can move between the environment, and it may become a commensal. And then within the host, you might exchange genes with other microbes present in the host and move between species. And so all of this together means that when we're considering molecular epidemiology of antibiotic resistance, it's a good idea to not just look at the epidemiology of the host, but you might need to consider the epidemiology of the antimicrobial resistance genes separately from that of the host bacterium. So now I'm going to stop a little bit and get a little bit into the nitty-gritty of exactly how we do molecular epidemiology of bacteria. So when we're looking at the host, there's been a long history of different types of ways of identifying bacteria. Because one of the first goals of epidemiology, of course, is to identify whether or not multiple people are getting the same bacterium, and identification of outbreaks. And one of the critical things here that has changed the most is how we identify the bacteria, and whether or not they're the same or different, identify clusters of related bacteria. So some of the major approaches that have been used over the years are serotyping, which was based on the reaction of surface antigens to antisera to identify bacteria that share serotypes. Then there was PFGE, was very common for a long time, where you would take the DNA, digest it with a restriction enzyme, and then analyze the sizes of different fragments produced by the restriction enzyme cutting at different positions in the genome. And that was essentially relying on sort of genome structure as well as mutation of individual restriction sites that might change where and how many different places where these restriction enzymes could cut. Then as the sequencing era got started, there was increasing use of multi-locus sequence typing, where they would pull, say, 6 to 10 housekeeping genes from a bacterium, where it's a gene that is commonly present across many different bacteria, sequence those and identify how many of those sequences are shared between different bacterial strains. And that leads to the sequence type, say ST131 is a common group of E. coli that are pathogenic. And that name comes from these original multi-locus sequence types. Now, increasingly, it's gotten to the point where these days it's cheaper to sequence a genome than to do all of this careful handling of an individual bacterial strain. And so that's where the newest technologies are heading, is whole genome sequencing. For a long time, we still will often go back and use the whole genome sequence to back predict the multi-locus sequence type, simply because that's a familiar terminology. Or you might try to back predict the serotype based on the genome sequence. But this is a very much still a moving target, but the field has increasingly been working to develop standards for using whole genome sequence data to evaluate bacterial relatedness, rather than trying to backtrack to these older sequence types. So whole genome sequence data, in theory, is the ultimate in identification of bacteria, because this is all of the genetic information carried by the bacterium at your fingertips in order to get the best picture of how closely related two different strains might be, identify clusters, outbreak clusters, and track back their epidemiology. Now, the problem is that it's so much information that we're still developing methods for the best ways to simplify this process down to the point where you can get a straightforward understanding of identifying related bacteria without necessarily having to go back and do all against all pairwise comparisons of your bacterium against every other bacterium in the database, which tends to be very, very computationally intensive. So there's been a couple of different approaches. Right now, the two most common approaches are sort of SNP, single nucleotide polymorphism-based techniques, and Kmer-based techniques. And so if you'll forgive me getting into the weeds a little bit, I'll give you just a brief introduction into what those two different type approaches are. So SNPs, or single nucleotide polymorphisms, in order to do those, you'll take a reference genome and then align all of your bacterial genomes to that and identify any place where your bacterium differs from the reference genome. And so this is just an example here. These were, I believe, Bacillus species. I pulled it out because they had a nice alignment here. The dots are any place where the base for that sequence is identical. And then here you might see these two bacteria have a C instead of a T in this position. So that would be a single nucleotide polymorphism. In another place, you have this group of organisms that have a difference, that have more differences with the reference genome. And then you can use those to identify strains that are more closely related or more distantly related to the reference genome and to each other. And you use those differences to reconstruct phylogenetic trees similar to the ones that Justin was showing to you 2 sessions back. And so you can either construct SNP-based phylogenetic trees or essentially use this database of SNPs to establish a database of reference alleles that is more similar to multi-locus sequence typing. And that's where the terminology is still in movement, where sometimes they're calling this SNP analysis. There's other people who are calling it whole genome, MLST. But some of the things, the advantages of SNPs was that it can be done using either assembled reads, or you can take your reference genome and your individual reads without assembling them, your individual short reads without assembling them, and look at read coverage at each position and call SNPs. The advantages is that it's high resolution. Some of the disadvantages of this is that your results are highly dependent on your reference genome selection. When you're doing this, when you have regions of the genome that are not shared, it becomes difficult to call. You tend to throw those out of the analysis. And then for groups of genomes, because you're throwing out regions of the genome that are not shared between organisms, it's restricted to the core genome. And so what I mean by the core genome is that when you look at bacterial genomes, this is, for example, a very old study, some of the early sequencing work on E. coli. And so they were comparing a uropathogenic E. coli species, the lab type K12, and an enterohemorrhagic strain 0157H7. And at the time, it was a bit of a shock because they sequenced these 3 different E. coli strains. They sequenced their whole genomes, and when they lined them up, they found that only about 40% of the genes across these guys were held in common. And each of these organisms had between 600 and 1,600 genes that were unique to each individual strain. And then when on an individual strain basis, about 60% of the genome was shared with both others. It's about 40% of the total genes that they recovered across all 3 genomes, but it's about 60% of any individual genome were genes that were shared between the strains. And then about 30% of each genome were genes that were not present in either of the other 2 strains of E. coli. And then how that tends to play out more broadly is that if you align, in this case, 5 different E. coli genomes, you'll have large shared regions, and then areas where there's higher rates of recombination, higher rates of mutation, higher rates of new genes moving in and out in between those. And so oftentimes when people start doing gene comparisons, they'll focus in on these core genomes, which tend to be, which in theory are the genes that are required for E. coli. They're less likely to be horizontally gene transferred. And so it's a better guide of the evolutionary history of the species as a whole, as opposed to the more recent evolutionary history of what genes it may or may not have picked up or kicked out again. But you'll also see approaches where they'll use the whole genome alignment and consider those accessory genomes in more depth, particularly if you're trying to distinguish between closely related strains. Oops. All right, so that's basically SNP analysis. It's also sometimes known as CG-MLST, which would be core genome multi-locus sequence typing, or WG-MLST, or whole genome multi-locus sequence typing. And I wanted you to get a feel for all of those different terminologies, which essentially follow the same basic analysis approach. So then the other major approach that is utilized are K-mers. So this is an approach that has gained some popularity because it makes for much faster, less computationally intensive searches. The downside of K-mers is that they tend to lose genomic resolution. So essentially what K-mers are, you take your genome and you chop it into DNA chunks of a specific length. In this toy example, we're chopping them into 4 base. We're chopping 2 sequences into 4 base K-mers. And then if you take this and you do 4 base windows of 2 similar sequences, you can see that all these regions where they're identical, you have shared K-mers. And then regions where they don't align, you can have some K-mers that are unique to one species or the other. These two get basically tossed out since TGAT already appeared in your sequence once, which is part of how you lose that resolution. But the upside is that it's much faster computationally. And then the next step tends to be some form of evaluating how many K-mers are shared between 2 bacteria. And the details of exactly how this distance is calculated and is highly algorithm dependent. In this particular case, for example, they went through and they found the species, they found the K-mers that were in common across the genus, and the species and the K-mers that were unique to each species. But use it to calculate a distance based on the number of fraction of shared K-mers and the fraction of unique K-mers. It's still called K-mers. In general, again, the length will depend on the program that you're using. Some of them will use a set length, usually something more like 18 to 36 base pairs. There's a few programs that allow for multiple different lengths of K-mers. But all of them follow the same general principle where you ignore alignment. You don't try to identify shared sequences by lining the genomes up and figuring out where they match. Instead, you just chop the genome up into little fragments and identify unique sequence fragments and shared sequence fragments. So the big advantages here is that it does not require a genome assembly or alignment, does not require a specific reference selection. It's much easier because it's faster. You can just do it pairwise for all combinations of organisms. And it tends to be a fast, less computationally demanding search process. It also tends in some ways to be less sensitive to repeat regions and other types of sequence artifacts. On the other side, hand in hand, some of the downsides of KMER is that it does have lower phylogenetic resolution compared to alignment-based approaches because it ignores this genome positioning. And it doesn't provide, you then would have to do a separate alignment to try to look at what the role of these different shared and differential regions are. Whereas with the SNP analysis, it's very easy to take your SNP profile and line it up with the genome annotations and identify what genes are different between the organisms. Here, if you want to know anything about how these two species differ, you have to then go back, assemble the genomes, line them up, and do the SNP analysis to understand what genes were changed to produce this different profile. And so as an example, the NCBI pathogen detection system I pulled up to show how this might look like, how a pipeline for doing this might look in the real world. So they've developed this whole pipeline to try and quickly analyze new genome sequences and identify clonally related isolates to aid in outbreak identification. And so the approach they started doing is they'll do a KMER-based assembly first. They do assembly first these days. They'll take their assembly, mask out repeats and low quality bases to make sure that they don't mess up their analysis. They'll do an initial clustering step using the KMERs, and then do pairwise SNP analysis with all the other organisms that fall into a certain KMER distance in order to then identify 25 clusters of clonal clusters with less than 25 SNP differences. Now that 25 SNP differences does not necessarily translate into 25 differences across the whole genome, simply because there's a lot of filtering steps before they declare that this is a SNP that they're going to use for identification. But it's a way of identifying clonal clusters, and then they'll, as part of the online algorithm, build a phylogenetic tree to show within cluster relationships between organisms. All right, so that's basically some of the ways in which they use whole genome sequence to identify clusters of related bacteria. But as we discussed earlier, we also have to consider antimicrobial resistance genes as moving in some cases independently of the host bacterium. So at that point, you have to identify your antimicrobial resistance genes. And so in terms of treatment, phenotypic testing with MIC is still one of the gold standards because it's one of the best ways to get a hold of the antimicrobial resistance phenotype of an organism. And that still does not, all of our best genome techniques are not always able to back predict antimicrobial resistance phenotype. But when we're looking at epidemiology, it's obviously very useful to understand the genetic basis of any antimicrobial resistance phenotype. So for a long while, we were doing PCR and probe-based methods, but again, as genome sequencing has decreased in cost, it's increasingly more cost efficient to just go ahead and sequence the whole genome and try to pick out the antimicrobial resistance genes. So when you're looking for antimicrobial resistance genes, you have a couple of different options. The hardest to find are essentially point mutations and spontaneous mutations, where the bacterium just needs to change one base or residue or amino acid in the protein that would then render the organism immune to the antibiotic. And as an example, quinolone resistance tends to be associated with spontaneous point mutations in a number of different elements of enzymes involved in DNA replication. In general, genomic techniques tend to be better at identifying antimicrobial resistance genes that are acquired by horizontal gene transfer. For example, beta-lactamases, where you might pick up a whole new enzyme that breaks down the antibiotic. And that tends to be easier to recognize because it's a known allele that is transferred between many different bacteria. But when you're doing a search for antimicrobial resistance genes, each of these requires sort of different search strategies to recognize. And again, these horizontally transferred alleles tend to be easier to identify than spontaneous point mutations. So I'm not going to get too far into the nitty-gritty, but there's two basic approaches to identifying a gene in the genome. Both are dependent on whatever your reference database is. The most exact tends to be BLAST, which is a homology search. So you take your DNA sequence, usually translate it into protein, and then compare it to a big database of protein sequences of other antimicrobial resistance genes. This can, in some cases, work fairly well with unassembled reads, but the cutoffs are sort of arbitrary or need to be set on a gene-by-gene basis. Because, for example, BLAST was really designed to recognize what gyre is. This protein is likely another gyre, for example, from E. coli, but you have a very different set of profile requirements to say, all right, this has definitely got the point mutation that we know can confer quinolone resistance in this organism, versus, oh yes, this is a CTXM ESBL gene. And then there's also HMM searches, where basically they build probabilistic models based on the fraction of, you take a database of known gene variants, identify how common individual bases are at each different position in the protein, develop this profile and use that to search your protein sequences for ones that match this profile. Nice thing is that when you're part of building this profile model, you pre-establish your cutoffs for what is going to be considered, for example, CTXM allele, and it tends to rank. It tends to place it's able to set greater importance on specific bases that we know are important for that particular function. But it tends to work best on assembled sequence and doesn't tend to work nearly as well on unassembled reads because you need the full protein sequence. And so to give an idea of how this tends to play out, this is the AMR Finder pipeline that they're using for the NCBI pathogen detection system. And basically, in order to identify antibiotic resistance genes, they throw everything at it. They'll do protein BLAST searches, they'll do Hummer searches, they'll do nucleotide BLAST-X searches to try to identify frameshift mutations. And they've got a huge protocol for calling how probable something is to be an antimicrobial resistance gene. And so then at the end, if we've still got a few minutes, I wanted to just put together to show you a little bit of how this might be used. So in this case, I'm pulling data from a study that my lab did of looking at ESBL Enterobacteriaceae colonization in community members in Athens. And so these were human fecal samples from healthy individuals. We isolated ESBL E. coli if they were present, and then looked at what types of ESBL E. coli we found. And so the first thing we did was we built a core genome SNP tree to identify how the sequences were related to each other. We also used some of the backtracking mechanisms to establish like sequence types for each of these things to compare to the databases of sequence types. We did some phylotyping. All of this was done in silico based on the genome sequence. And then we also did pairwise SNPs of each isolate with a second isolate that we obtained, if we'd obtained one when we brought our subjects who were ESBL positive back 3 months later, looked to see if they were still colonized by ESBLs, and looked at how that isolate was related to the original one. And so as you can see, we found that there was a diversity of ESBL E. coli present in our subjects. In general, most of our repeat isolates were somewhere between 50, somewhere around 50 SNPs differences between that guy and the first isolate that we got. Then looking at, for example, we went and looked for ESBL genes on what ESBL genes each of these isolates might carry. We found that basically all of them carried at least one CTXM type beta-lactamase. But as you can see, based on the stars, quite a few of them were carrying those on mobile genetic elements, either transposons or plasmids, and in many cases they were plasmids. We also looked for other antimicrobial resistance genes that might be present on these isolates and found quite a few of them using that AMR finder program that NCBI uses. And then we found a few interesting things that I wanted to point out just because they're examples of how this might how some of this horizontal gene transfer in bacterial epidemiology might relate. Where, for example, we had 2 isolates from 2 different individuals that were very nearly clonal. But when we looked at their antimicrobial resistance profile overall, it was quite different. And what we found there was that both of these isolates were carrying near identical INCF plasmids that had, in this individual, both your beta-lactamase gene here in purple, and then a whole bunch of other antimicrobial resistance genes down here in red. On the other hand, for the isolate obtained from subject 486, lacked all of these, had apparently lost those transposons, or this guy had gained them, and instead just had the beta-lactamase gene and nothing else stuck inside their plasmid. So this huge group of antimicrobial resistance genes either hopped into one of them or hopped out of that one using transposons, changing a vast array of antimicrobial resistant phenotypes for these 2 isolates that were otherwise very, very similar. We had another case where we had one subject who we brought in after we initially tested, and they had one type of E. coli that was carrying a CTXM1 gene on a plasmid. When we brought them back 3 months later, we were able to isolate an ESBL E. coli again, but that ESBL E. coli turned out to be from a very different group of E. coli. So these were not the same bacterium at all, but they actually shared a near identical plasmid that included the CTXM allele. And so what we think happened there was that probably both of these E. coli were co-resident in the gut for some period of time, and one of them passed the plasmid on to the other one, which then took over in the gut. And so as you start to look at antimicrobial resistance epidemiology, as I was putting this session together, I worked to bring together a variety of different perspectives. So next week, you've got a break. We had to move things around to accommodate all of the different speakers' schedules. But on the 19th, you're going to be hearing 2 talks, one from Elizabeth McMillan, who is she was a UGA grad student working in the lab of Dr. Jonathan Fry on in the the poultry microbiological safety unit, trying to understand antimicrobial resistance and salmonella found in poultry. She's about to start a new position as a research scientist at the USDA, and I brought her in because I thought it'd be useful to hear not just from people working on humans, but it'll give you a chance to understand how food safety scientists have been approaching epidemiology of antimicrobial resistance. And then Michael Anderson and Tatiana from Department of Public Health are also going to be speaking on the 19th. And then for the 26th, for our guest speaker, I invited Latanya Logan, who is a faculty member who recently moved to Emory, who's been doing some really interesting work on ESBL E. coli, as well as the role of community spread and environmental spread of ESBL E. coli in influencing antibiotic-resistant infections in children. So with that, I'll take a break and see if anybody has any questions.